Hi, my name is Schapira, and today I will be presenting my project on screening for new pockets in WDR proteins. This project was conducted at the Structural Genomics Consortium at the University of Toronto. Before getting started, I would first like to mention that this entire project was done using ICM Pro, a software developed by Malsoft. So first, why analyze WDR proteins? WD40 repeat domains, also known as WDR domains, are protein interaction hubs involved in a diverse array of signaling pathways. WDR-containing proteins are one of the largest and most disease-associated protein families, but potent drug-like ligands have so far been reported for only 3 out of nearly 400 proteins. A great example of discovering new bonding points on a WDR protein, like my project hopes to replicate, is with the EED protein structure. Displayed is the original protein structure with no good binding pockets. It is important to note that the entirety of this project is focused on the middle top part of WDR proteins, as shown. Next, it has been experimentally proven that a more druggable pocket can be obtained with a new conformation, now shown. Using this new conformation, a ligand can then be bonded to the protein structure. Looking at this change in more detail, 2 specific residues had to move for the binding pocket to form. These residues are highlighted with the original conformation in gray and the new conformation in light blue. Furthermore, the new pocket shown on the rightmost graphic shows the original residue's placement blocking the pocket from being formed. Similarly to the EED structure just shown, the WDR5 protein structure has had a similar discovery. Once again, the top middle residues of the protein could be moved to form a better binding pocket. Once again, the residues that moved the most are highlighted, with the original one in red and the new conformation in blue. Based on the 2 previous proteins, the goal of my project was to write a script that would find new binding pockets in other WDR proteins, just like the EED and WDR5 protein structures. To test my code, I ran it on the original structures of these 2 proteins, trying to replicate the new conformations previously shown. Once my code discovered the same pockets successfully, I ran it on the entire WDR protein family, composed of 361 human proteins, and analyzed the results. Before going into more detail, let's first look over a general overview of how my script is constructed. The first step is to simply load the protein structure. Then, the amino acids forming the WDR structure are isolated. Next, the side chains blocking a potential pocket are identified. As a reminder, this project's focus is only on the top middle part of these proteins. Once identified, I used a Monte Carlo simulation to try moving the residues. I will explain this simulation in more depth later on in this video. Lastly, I analyzed the results, and a website showcasing the findings will be made. Now, looking at the script structure in more depth, after loading the protein and isolating the WDR part of the structure, the next step was to identify the specific residues blocking a potential pocket. First, because the side chains are way more flexible than backbone atoms, the first step was to find a pocket that does not overlap with any backbone atoms. This allowed the script to try to look for binding pockets without having to move the backbone atoms far. On the left is the original EED protein structure, and on the right is the same structure after simplifying all side chains to alanine structures. Simplifying the side chains like this essentially only leaves the backbone atoms left, which are then used to find a pocket that warps around the backbone atoms. As a note, this strategy will not work on all protein structures because some structures have backbone atoms blocking the middle of the protein as well. After finding the pocket that goes between the backbone atoms, the pocket was then overlapped with the original full structure. This shows us exactly what side chains are blocking the pocket from being formed. In this case, on the EED structure, this one residue is blocking it. Taking a step back, it is important to recognize that looking for pockets in a structure made only of backbone atoms will return tons of pockets. I superimposed another WDR structure on top of the original structure as shown. This new WDR structure has a constant atom placed above the top middle part of the structure, colored in blue. The script then runs through all the pockets and chooses the closest one to the blue atom. This blue atom is always above the middle of the structure, which makes it a useful reference point throughout the entirety of the script. These pockets were found using the ICM pocket finder with a tolerance of 0.3. However, when running the same script on the EED structure calculated by AlphaFold, it only finds a pocket when the tolerance is set to 4.6. So, when running through the AlphaFold database, I decided to follow that tolerance. Now that the right pocket was identified, the obstructing side chains, as well as the side chains around it, were isolated. The next step was to run a Monte Carlo simulation on those side chains. A Monte Carlo simulation randomly runs through different conformations that a group of atoms can take and only accepts the conformations with the lowest energy levels it can find. Each Monte Carlo step generally accepts the lower energy conformation, but higher energy levels are also accepted with a low probability to avoid getting stuck in a local energy minimum. This means that by running through enough random conformations, it will eventually find the most optimal ones and save them. A low energy level represents a higher chance for the conformation to actually occur. Unfortunately, randomly moving the selected residues does not necessarily make them move in the positions wanted. To help push them out of the area with a potential pocket, 3 unrelated atoms were added. These atoms cause a clash with the side chains, which, in turn, create a spike in energy level. The Monte Carlo simulation then tries to solve this spike in energy by moving the side chains away from the added atoms. Displaying the middle side chains in a space-filling model helps show the clashes between the atoms. An important note to recognize is that because some pockets are deeper than others, 3 different Monte Carlo simulations are run, starting with the topmost atom being added and a slightly deeper atom each time. This allows for cases with a shallow pocket to still be successful by running the simulation with only the shallowest atom. In order to consistently add the 3 atoms in the middle of the backbone pocket, the script used the average location of the backbone atoms around the pocket. To identify different parts of the backbone atoms, the script took the backbone atoms at increasingly far distances from the constant atom placed above the middle of the protein. These distances were 10, 14, and 17 angstroms respectively. The selected backbone atoms must also be within 5 angstroms from the pocket. Then, as mentioned before, the average location of these groups of atoms determines the location of each added atom. As a side note, 10 backbone atoms were needed for the atoms to be placed. This ensured that the average location was actually in the middle of the pocket and that the selected pocket was the right one. Although this strategy places the added atoms along the center of the pocket well, it sometimes places them relatively far apart from each other, which leaves gaps that side chains can bury themselves in. In order to plug any gaps between these added atoms, this small block of code creates a vector between two added atoms at a time and brings them closer to each other in increments a tenth of the initial distance between them. The same block of code was copied and applied to all three atoms. After adding these atoms and running a Monte Carlo simulation again, it was clear that adding the three atoms helped significantly. The WDR5 protein worked relatively well, but the EED structure could still be improved on. In green is the experimentally proven EED conformation that I am trying to replicate. In beige is the original EED conformation, and in purple is the new conformation from the most recent Monte Carlo simulation. This new conformation opened up a small pocket shown in blue. However, the goal conformation has its residues much further away and a much bigger pocket, which means that there is still room for improvement. Two possible solutions that I tried for even better results were to move the added atoms up, which would, in theory, push the side chains up even more. Unfortunately, this did not work very effectively, as well as the solution being very specific to the EED protein, while I am planning on analyzing all WDR proteins. The other solution was to allow the Monte Carlo simulation some control over moving the backbone atoms. The reasoning for this is that the backbone atoms in the goal conformation moved slightly too, as shown in the red circle. Finally, this solution returned a very similar conformation to the goal protein structure. In the right graphic, the green residues are from the goal structure I'm trying to replicate, and the orange residues are from the new Monte Carlo simulation. Although these results were promising, adding the backbone atoms greatly increases the number of variables the Monte Carlo simulation needs to compute, as seen as in the bottom left graphic. This in turn causes the simulation to get inconsistent results and requires more computational power. As a reminder, a Monte Carlo simulation tries to run through as many conformations of variables it can in a somewhat random way. So increasing the number of variables increases the number of conformations needed to be tested. Lastly, to solve this final problem, the three following steps were conducted in order on each of the three atoms at a time. I first started by only selecting the side chains that are clashing with the current added atom, instead of all the side chains around it. Then, I randomized the identified side chain's position before running a Monte Carlo on it, the side chains around it, and the backbones further around it. I then replicated the last two steps 7 times, each time starting with the original structure and saving the results. This solution moves the obstructing residue before starting the simulation at all, with the hopes that one of the 7 runs already starts with an open pocket. The results of this solution were very similar to the conformation I was trying to replicate. The last step before running my script on the WDR protein family was to choose the best conformation out of the many that each run found. The graphics so far have always shown the best results of each Monte Carlo simulation. However, the script needs to be able to choose these conformations on its own. I ended up using 3 conditions that slowly filter out conformations and ordered them in order of least computational power required. First, I made sure the energy level was negative. A higher energy level represents a lower chance of a conformation to occur. Next, I made sure the energy strain on the residues around the pocket were below 3 kilocalories per mole. And lastly, of the conformations left, I took the one with the best pocket D-lid. D-lid is a value calculated by the ICM software to quantitatively rate the druggability of a binding pocket. Now that everything was set up, I ran my script on the AlphaFold WDR protein database using Compute Canada's servers. It would also be useful to run this script on experimentally found WDR proteins. However, my time limit on this project limited me to the AlphaFold structures. I chose to start with AlphaFold because of how extensive their library is. Once my script was done running, 28 proteins were found to have a new pocket and 15 proteins had their pockets improved. Lastly, a website showcasing my findings will be made. Shown is a page of the website displaying the first protein with a new pocket. This is the original protein structure. And this is the structure with a new pocket. The website also shows the side chains that moved the most in order for this new pocket to form, as well as a comparison with their new conformation. Thank you for your time, and thank you to Matthew Schapira and Avian Rovers for their supervision at the Structural Genomics Consortium at the University of Toronto throughout this project.